Hello, everyone. Thank you very much that you are all here. It's a really pleasure to see so many experts in the ultrasonic vocalization. Today, I'll be talking about exploring the ultrasonic vocalization role in reward-related memory and goal-directed behavior. The first part will be not about the USV, but the second part will be you will see that some of the USV researchers also included in the presentation. So let's start. So the formation of memories is a complex multi-scale phenomena, especially when it involves integration of information from various brain systems. Spatial memory is defined as a brain ability to encode key features of the external environment and to navigate within the boundaries of the mental representation, also known as a cognitive map. In case of episodic memories, their spatial context can be, among others, linked to the emotional state during learning. In our experiment, the rewarding incentive was the amphetamine. We used those 1.5 milligrams per kilogram IP, so for conditioning. Amphetamine is known to activate reward system and induce emission of 50 kilohertz band ultrasonic vocalization, which are a marker of affective appetitive states in rats. All right, so first of all, I will tell about the pipeline of the experiment. So this is the testing cage, the training and testing cage. On the top, we've got the special cues. So this is a model. This is real one. We've got the microphones in each corner, so it's a kind of open field with the four corners. Yes, during the training, the corners are closed, and during the test, the corners are open. Yes, so the animal can explore the whole cage. This is the pipeline, the training and test. So how it looks like. So during habituation, the spatial cues are turned off. So the animals get the habituation to the cage. So all doors are open, and then we've got the training phase, and there's six training days. So on the day 1, it's receive the saline administration in the corner C1. Then on the day 2, they got the amphetamine administration in the corner C3. Then on the day 3, they got the administration of the saline in the corner C2. Then they go back for the second dose of the amphetamine in the corner C3. On the day 5, they got the last saline injection in the corner C4. And on the day 6, they got the third amphetamine injection in corner C3. Then we divided the animals to 2 groups. So it was the recent recall group and remote recall group. The only difference between those groups were the hold-off period. Yes, so it was the 13-day delay. So the recent guys went to the testing days on the day 7. And day 28, those remote recall group got the testing on the day 28. Sorry. Yes, and we got two phases of testing. We got the first entry, and during the test, all doors are open, so animal can explore the environment, and the special cues are, of course, on. And we've got the first entry, so this is the recall process, but they didn't get the amphetamine, so they got some kind of conflict. Yes, so they got some kind of conflict because, oh, wow, where is amphetamine? Okay, so then after 20 minutes. They got the second test, so we believe that it's a reconsolidation process. So they reconsolidate their memory, yes, using the memories from the first entry, and explore the cage in the second entry. Why we did two tests? Because we used the cellular compartment analysis of temporal activity by fluorescent in situ hybridization. It's a catfish method. When you look on this graph, yes, here you've got the ARC transcription related to the first entry. Then this is the point where are we looking at? Yes, so from the first entry, we've got the Homer 1 transcription related to the first entry. So this is those green dots. Yes, and from the second entry, we've got the red dots. So this is this one, and it's related with the ARC transcription. We synthesize the probes, yes, and do the reaction to see which neuronal population was active in the first and the second entry, and which population were active in the first and the second entry. So we've got inactive nucleus. Here is a nucleus, so we can see that it's inactive. This one was active in the first. Exclusively in the first entry. This one was active exclusively in the second entry, this one. And this one, for example, were active in the both. Yes? So we got some kind of colocalization of the activation. So we know that this neurons were active in the first and the second entry. And this is this element of the CA3. All right, so where is the engram? Yes, the engram of the recent and remote. Yes, so where we try to find it. So the engram was measured in the prelimbic cortex, infralimbic cortex, CPU, nucleus accumbens, shell, and core, and the hippocampus, the CA1, CA3, and DG, the gyros. Of course, in the amygdala nuclei, and also in the BTA. All right, so this is the overview of the whole experiment. So as you can see, this is the first entry, so the green ones, and the second entry, the red ones. All right, so the aims was to investigate the mechanism involved in the interplay of spatial memory and reward processing systems. Understand how this interplay influences emotional context perception, and utilize machine learning to pinpoint the brain circuits responsible for reward-seeking behavior guided by spatial cues. If we compare those two groups, looking only on the behavior, we made a lot of parameters, so almost 20 parameters of the behavior, and we found just the differences in rearing. Those guys from the remote group got the decreased rearing, and got the time in any corner. So only the time in any corner was the difference. But what was happening in the brain? So remote recall involves smaller and more consolidated neuronal subpopulation in centers in certain structures. So if you look under any active, so it's a Homer or ARC, yes, and so this is the any active. You can see, okay, the green is a recent, the color, orange. Okay, let's say orange is a remote group. So those guys after the hold off, and you can see that. We have drop down of the activity activity in many brain structures in the remote group, but we've got some kind of specialization of those neurons. So we got the specialized neuronal populations in few of studied structures. Especially, you can see that it's in the BTA. We've got something in the nucleus accumbens, but we've got two guys. I will show you that consolidated very much faster than another group, and we've got the same situation in the DG, the data gyrus. So I would like to that you. So we've got the overall drop the activity of neuronal activity and specialization of the neuronal specialization. So recall of the remote memory has a district activity pattern from recent. We use the Boruta algorithm that machine learning algorithm to differentiate the groups. So we would like to see where the engram is going. So where is the switch of the engram? So how to read it? I know that it's probably most of us are familiar with this, but I'll tell you how to read it. Okay, so here we've got the recent, so the green ones, and remote groups. Okay, this one is recent, yeah? So the greens is recent, and the orange one is remote, so after the hold-off period. And you can see that, those groups divided in the brain. So you can see that the recent rats got the activity of the high activity of the dentate gyrus, hippocampal dentate gyrus, and also the VTA in the first entry. But those remote, so those rats which got delayed 40 days delay, we can see in this in the brain some kind of switching the engram. So the engram is somehow transported to other structures, but not so we can see that we got the colocalization in the ventral tegmental area in the group of remote animals. We got some engram in the prelimbic cortex, but we've got something in the second entry exclusively in the CA3. As you can see in the recent group, so those guys which were not have this delay, we've got the CA3 active in the first entry. So they use the CA3 on the beginning, yes, but when you look at the remote group, yes, oh, okay, I forgot to say something. The blue is a low, and the green is high. Okay, that's how to read it. Yes. So those guys from the recent group that got the activation of the CA3 in the first entry, but in the second entry, after process of reconsolidation, those rats from the group of the remote group got the activation also of the CA3. So I hope you understand how to read it. We can see that we've got some clusters of structures. Yes. So we have also activation of lateral amygdala in the first entry in the recent group. All right. So as I said before, we didn't find any differences in the behavior actually between those animals. But high memory remoteness independent variation of the nucleus accumbens is explained by behavior. We used the correctness score. So this is the correctness parameter, which means the time spent in the conditioning corner to whole time in the other corners, in the all corners. Yeah? So this is, we call it correctness. But okay, let's start with this. And how it looks like. In the first entry, we've got this is the correctness score. Of course, it's a rank. Yes, it's the rank. So this green one, and you can see this is this parameter. So this is the green one, and you can see that this parameter correlates or is related with the activation of the nucleus accumbens core and shell in the first and the second entry. No matter if it's remote or recent group, okay? And also we've got the activation in the CA3 CPU. We've got some relation in the retrospinal cortex, and yeah. and colocalization, relation with the colocalization at CA3 and nucleus accumbens. But what happens in the second entry? So they got some kind of conflict in the first entry, so they didn't get the amphetamine. So I don't have the amphetamine where it should be. So they got the process of reconsolidation. And what happens in the second entry? So this is the same. So this is the correctness score. Yes, the green one. If it's more green, it's more correct. Yes. And we can see that there is another thing. So we got the nucleus accumbens core colocalization. So we got the specialized neurons in the nucleus accumbens. VTA, we got some relation with the VTA in the first entry, but you can see that here are the remote and here are the recent guys. And we get some relation with the central amygdala in the first and any, yes, because the any is first or second. So this heat maps looks like this. All right, so we can see that the activation of the nucleus accumbens and CA3 and also CPU was related with the correctness in the first entry, but in the second entry there's more with the amygdala, but we still have nucleus accumbens. All right, so the strongest interstructural coactivation occurs in the second entry. So here are both. Okay, so this is the correlation, the Spearman correlation between structures. Of course, we didn't use 100 animals because the experiment should last 4-5 years. Yes, but it's about 20 animals. So we got the positive correlation in the nucleus accumbens shell, between nucleus accumbens shell and core, and also between the nucleus accumbens shell and CPU, and also the trivial positive correlation between the CA1 and CA3. When we look on the first entry, so we can see that we've got some crosstalk between basolateral amygdala, central amygdala, lateral amygdala, and between nucleus accumbens core and shell, and of course in between CA1 and CA3. So this is the first entry. When we, but the first entry means that it was those dots with green and those dots with the green with red. So this is the first entry. When we look exclusively on the first entry, so only the green dots, yes, so activation of the neuronal population in the first entry, we can see that there is additional correlation. So we still get this crosstalk between basolateral, central, and lateral amygdala. So the positive correlation between those nuclei of amygdala, but we get also the positive correlation between retrosplenial agranular and granular cortex. And what happens also with the prelimbic cortex? Prelimbic cortex negatively correlates with the central amygdala. What happens in the second exclusively, so after reconsolidation process, is, as you can see, this is the strongest interstructural coactivation. Yes, so we've got this crosstalk between nuclei in the amygdala nuclei, so basolateral, central, and lateral amygdala. We've got the positive correlation between CA1, the hippocampus, and CA3, and we've got the positive correlation also between the CA3 and agranular retrosplenial cortex, and agranular retrosplenial cortex positively correlate with the granular retrosplenial cortex. But we've got also coactivation of the CA3 and DG. What else? You can see that prelimbic cortex coactivate with the infralimbic cortex, and it's a positive correlation. And the red one is a negative correlation. So we've got the prelimbic cortex negatively correlates with basolateral amygdala, and hippocampal dentate gyrus. Okay, this is the summary of this part, this results. So here on the right, you can see the differences catched by Boruta machine learning algorithm between recall groups. Yes, so we've got a few of structures that differentiate those animals. It's CA3, prelimbic cortex, DG, dentate gyrus, granular retrospinal cortex, but exclusively in the second entry, and also the colocalization of the in the VTA, dentate gyrus, and lateral amygdala. When you look on the correctness score in the first entry, we can see that nucleus accumbens was the structure what was most differentiating or get the relation of the correctness of this in the first entry. But we also get the CPU, the CA3, and the retrospinal granular cortex. But in the second entry, as you remember on those heat maps, we've got the relation with the infralimbic cortex, nucleus accumbens core colocalization, CA3 colocalization, and CA3N. So, yeah. All right. So, but what about the ultrasonic vocalization? Yeah. So we skip from the neurobiology to talk about the ultrasonic vocalization. Of course, we got the microphones in all corners, and we recorded the ultrasonic vocalization during the whole experiment. We used to analyze. We used our software RATREC 7.5. We developed it 10 years ago, and something like this. This is not from this experiment. Those vocalization is not from the experiment. This is from the other experiment, the social interaction after isolation, social deprivation. All right. So, what are the differences between those groups? So, there are no differences between groups. So, we cannot see the switching the n-gram as from the recent to remote group in the ultrasonic vocalization. So this is the whole of emitted sounds during the training. The first day, second day, third day, fourth day, fifth day, sixth day, and this is the testing day. So you can see that there was very less of vocalization during the exploration. That we got the informations from the brain, so we know which population of neuronal population were active in the test. So we did the machine learning Boruta to see if there is any relation of the vocalization with the activation of neuronal activation during the tests. And we found, okay, so this is the remote, now it's pink, yes, and the blue, the recent ones, and episode. This is not the number of episodes, this is the rank, so we don't look at this. Right, so you can see on the green that if it's more green, then you have more vocalization. So the only structure that were related or catched by machine learning was CA1. It was exclusively in the first entry. But when we look on the training days, so we try to understand why only the CA1. Of course, if we increase the n so to 100, so there we will catch more. But yeah, we didn't did that experiment with 100 animals, and we found that the training day, so the third training day, it was the saline injection in the C1. Was related with the infralimbic cortex. So if we have infralimbic, was related with the activation of the infralimbic cortex in the test, after test. So here you can see that those green those green ones was the vocalization from the training, the day 3, and it got the relation in the infralimbic cortex. When you look on the day 5, it was also the vocalization in the C4 corner. So it was also the saline one, and we can see that we have something strange for me, but okay. Maybe you get the answer for this. So here is the rank of the episodes emitted during the day 5, and we can see that those those animals which were vocalized during this training day was related with the decrease activity of the nucleus accumbens during the test. So when I look on this results, so what, how it's happened? All right. So and we got the relation of the training test vocalization on day 6. So it's amphetamine administration, and we got the relation of the ultrasounds with the retrosplenial cortex, especially in the first entry. All right. So I got two more slides. So we look on the behavioral data, so we correlate. This is the correlation, just correlation. So the red one is a positive correlation, the blue one is a negative correlation. So we've got the all training vocalization. We've got the correctness score. So we can see that those vocalizations were consistent within animals. So here we have high vocalizers, yes. Here is the animals that vocalize less. And what happens? The correctness score, so this one was get the negative correlation with the ultrasounds emitted during the training. And it looks like this. Okay, so if those animals which were vocalized a lot got the low correctness score. This is the whole message. So in the conclusion, so the remote group of observed neuronal activity is generally lower, but more specialized. Yes, so we get this process of consolidation. The memory engram switch is not visible in the behavior. Behavioral outcomes induced by memory recall can be heavily modulated by independent factors, including individual variability, especially when emotional processing is substantially involved. Nucleus accumbens is a key structures enforcing action, while the amygdala is likely an integrator or modulator. Strong emotional processing after administration of amphetamine or anticipation decrease the correctness value. So the higher activity to amphetamine negatively correlates with the correctness parameter. Individual variability is a crucial for development of effective intervention, both experimental and therapeutics. Of course, after each experiment, we've got more questions than answers. So this is the generated questions. So do rats vocalize more after amphetamine administration are less prone to addiction? And those have a lower visitation of the conditioning corner, or are highly vocalizing rats conditioning to the entire cage and not only to the specific location, the corner of the cage associated with reward. All right, I would like to thank a few people, Miron Kursa, which will do the computation, Tomasz Jaroszewski, Wiktoria Karbicka, Monika Falińska, Konrad Danielewski, Urszula Włodkowska, and Krzysztof Piwoński, and of course Rafał Czajkowski, the head of our laboratory, Emilia Rymak-Kozicka, and Stefan Kubik. Okay, thank you very much.
Thank you, Adam. It was almost perfect on time. So we have a time for a couple of questions. I will just check. So maybe that's before the break.
All right.
Yeah, late. Yeah.
All right. Yeah.
Yes. So questions, please. Just stay here.
Yeah.
Yeah, it's better here.
Okay, so people can hear. Okay, thanks very much for this enormous amount of data. So I'm sure I didn't catch it all, but one of the questions I was wondering, looking at your experiment, which is really brilliant, is one of the many questions, right? Is whether the animals that shows the highest activity in the nucleus accumbens, whether they are expressing frustration for not, because it's a place preference experiment, right? So in the Schultz experiment, where the activity of nucleus accumbens is dropping, actually not increasing, but dropping, when animals expect receiving a reward and didn't get it. So do you think this could hold for that?
We don't know yet, actually, because the nucleus accumbens was active in the first entry, so it was just the moment that they got their recall, yes, so the recall of their memory. So the searching, so the activity of the nucleus accumbens in the first entry, this was just recalling, yes. In the second, yes, there were in the second entry there was a colocalization, activation of the colocalization, but it was correlated with the correctness score. So now I really, but we have to do another experiment, because we don't understand why those animals which were high vocalizers got the low correctness score. Yes, maybe they are. It's hard to be addictive when you are very happy, or if you get the activation of the higher activation of the reward system. We don't know.
They just express their, I don't know. It's
frustration.
Maybe they just express their frustration. It's not, because place preference is not addiction. Is the memory of the sensation that they have while being in this context. So maybe they are just sort of frustrated, and they have a maybe better memory of the feelings, or they rely more on their feelings than on their maybe there's episodic memory. I'm just making out, but maybe the episodic memory is more based on their inner feelings than cognitive sensation or cognitive appreciation of the situation, of the context.
Yeah, it might.
Probably Paul has more other things.
It might be quite right. Should explore this subject more.
Yeah, that was a very complex talk, I have to say. Very impressive. Maybe I'm just going to say the same thing as Sylvie in different words. When an animal has been conditioned with amphetamine in one corner and then goes there, is the animal, do you think, experiencing an amphetamine-like state, or is it more out of optimism? I've been rewarded there before. If I go there, maybe I'll get something good. But maybe when they get there, there's nothing good to be had. So what do you think about that? And also, does the corner where he first goes predict where he would stay if you allowed him to stay? Because if I understand correctly, after the first entry, he's removed from the apparatus. Yeah. So there's my question.
All right. I'll tell about I will try to answer for the first question. So if you think they all right. We know probably as though those vocalizing guys are more optimistic. If they are okay, maybe they are more optimistic. So when they are going to this corner, yes, if I understand, they got the higher optimism, but we've got the parameter of correctness was much higher in the animals which were low vocalizers. So I think that the optimism in this case is not the case. So maybe they predicted because, or they got some kind of I think it might be that this corner is related with for example, place cells, yes, in the hippocampus, and when they are in this corner, which was the corner where the reward was administrated, as they got the activation of the place cells, and there's connection between place cells and the reward also system, and so the nucleus accumbens, and we can see this in when we were analyzing the vocalization, because the vocalization of the vocalization during the training was much higher when the animals were close to this corner, to this conditioning corner where we will give the benefit. Yes, yes, this is the.
We'll continue at coffee.
Yeah, sure. Thank you very much. Thank you, Paul.